Reaction time with Dave. We're about to get started. Yeah. I love this thing. Okay. Hello, everyone. Welcome to another episode of Reaction Time with Dave. Once again, I got with me my main man, Jack Phelps. So, today is a special day. A very, very special day because there's something I wanted to know. And I saw I was on the internet, I was watching the TV, I'd be on the YouTube, and I was like, what's this crispy stuff? And I was like, and then I found out it was actually called CRISPR, so I put the CRISPR, and it showed me everything that was crispy. So I'm like, chicken. And I'm
Number
like,
9.
Number 9. He says number 9. So, but no, what we're talking about is, what I need to know is, what is CRISPR? Like, what is it? CRISPR, CRISPR. Do you know?
To be honest, it's very difficult. It's complex, and it would definitely take more than just us searching the internet trying to figure out what it is to tell you what it is.
Okay, so I guess what we gotta do is we gotta take a look at how these extra crispy pieces of chicken, we gotta take a look at how the CRISPR, or what is CRISPR, or well, let's look at all of it. What is CRISPR? What does it do? Who it turns you into if you eat the chicken. I'm joking. So we're gonna go ahead and we're gonna take a look at CRISPR, everybody. Let me take a look at this. Okay, it says CRISPR, CRISPR. It says this article is about the prokaryotic antiviral system. I know that's a big word. We'll tell you what that stuff means later. CRISPR, an acronym for clustered, regularly interspaced short palindromic repeats, is a family of DNA sequences found in the genomes of prokaryotic organisms such as bacteria and archaea. These sequences are derived from DNA fragments of bacteriophages that have previously infected. Like, listen, that's this.
Right. This will bore anybody.
Whoa.
As informative as this is, and you could develop a lot of knowledge. If a person landed here, they would be immediately turned off to what CRISPR is, and that cannot happen. This is generally what people do. They will accept Wikipedia for college student reports as sources. Someone reading this wouldn't really go too much further. So this is how we landed on it, but we are due diligence for everyone.
We're gonna bring you into the forward. CRISPR is gene editing technology in theory.
Yeah.
You know what I mean? It uses it to attack pieces of your DNA, and they also use something called Cas9, which is an enzyme that uses CRISPR sequences, and we'll go into that hand in hand. So we're gonna tell you what Cas9 is, and then we're gonna jump back into what the whole thing is for you. So we're gonna go over here and show you Cas9. Cas9, CRISPR-associated protein 9, formerly called Cas5, CSN1, or CSX12, is a 160-watt kilodalton protein which plays a vital role in immunological defense of certain bacteria against DNA, viruses, and plasmids. Basically, Cas9 can stop things from invading the DNA and causing a virus, like genetic viruses we're talking about, like birth defects, things like that. That's as simple as we can explain it to you right now without you guys getting crazy. So we're going to jump into a little video here, and it's going to explain to you. We're going to got a couple examples and a couple things for you, but this is going to give you the base, and then we'll dumb it down for you. And then we'll go into what it could be, what is going on, and what it is. So let's go over here and check out genetic engineering. Woo, woo!
Computers would soon take over everything, from shopping to dating and the stock market. That billions of people would be connected via a kind of web. That you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality, and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. Where it came from, what we're doing right now, and about a recent breakthrough that will change how we live and what we perceive as normal forever.
Genetic modification.
Humans have been engineering life for thousands of years. Through selective breeding, we strengthened useful traits in plants and animals. We became very good at this, but never truly understood how it works, until we discovered the code of life, deoxyribonucleic acid, DNA, a complex molecule that guides the growth, development, function, and reproduction of everything alive. Information is encoded in the structure of the molecule. 4 nucleotides are paired and make up a code that carries instructions. Change the instructions and you change the being carrying it. As soon as DNA was discovered, people tried to tinker with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to get a useful plant variation by pure chance. Sometimes it actually worked too. In the 70s, scientists inserted DNA snippets into bacteria, plants, and animals to study and modify them for research, medicine, agriculture, and for fun. The earliest genetically modified animal was born in 1974, making mice a standard tool for research, saving millions of lives. In the 80s, we got commercial. The first patent was given for a microbe engineered to absorb oil. Today, we produce many chemicals by means of engineered life, like life-saving clotting factors, growth hormones, and insulin. All things we had to harvest from the organs of animals before that. The first food modified in the lab went on sale in 1994. The Flavor Saver tomato, a tomato given a much longer shelf life via an extra gene that suppressed the buildup of a rotting enzyme. But GM food and the controversy surrounding them deserve a video of their own.
Modified organism.
In the 1990s, there was also a brief foray into human engineering. To treat maternal infertility, babies were made that carried genetic information from 3 humans.
Okay.
Yeah.
So they said genetic information from how many people does it take to make a baby? One. 2. So are they injecting? Look, there's a little sperm. There's a little. I'm saying what's going on here. He said genetic information from 3 people. So we're already playing with things before CRISPR. We're already playing with things of this nature, it seems. Cytoplasm with healthy mitochondria.
Making them the first humans ever to have 3 genetic parents.
3 genetic parents.
Today, there are super-muscled pigs, fast-growing salmon, featherless chickens, and see-through frogs. On the fun side, we made things glow in the dark. Fluorescent zebrafish are available for as little as $10. All of this is already very impressive, but until recently, gene editing was extremely expensive, complicated, and took a long time to do.
And that's what we're talking about. Extremely expensive. Extremely expensive. Like, this research was in labs. This is hundreds of millions of dollars being spent on this. There's people with degrees. So we're going to go to how CRISPR, lets you edit stuff. And as you see up there, CRISPR for Dummies, I told you we're going the way where everybody's going to get a little bit of the love as far as, because these are big words they're using. So we're going to make it in layman terms, because not everything is even understood by the scientists. So how could you possibly expect us to understand it all as well?
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science.
It's the cheapest.
But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses two main components. The first are short snippets of repetitive DNA sequences called clustered, regularly interspaced short palindromic repeats, or simply CRISPRs.
That's a
The
lot to
second
take
are
in.
Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors.
Oh, CRISPR
When a
associated
virus
proteins.
invades a
That's where
bacterium,
Cas9 comes
Cas
from.
proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA.
Okay.
This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the
Well, are
gene
we
they
all
want to
errors?
edit and attach it to Cas9.
What is deemed as
Like
an
the
error in
viral
this?
RNA in the
people
CRISPR
deciding
immune
this.
system, the guide RNA
decide?
directs
You
Cas9
those
to the
people
target
are
gene,
errors.
and the
So
protein's
we
molecular
have this
scissors
lab, we have
snip
the host.
the DNA.
So we got, and we're giving you, this is another deep in this. How do we know what DNA is good and what DNA is bad? On there I saw there was human genetic, I saw traits under one of those squares. It said human traits. So then you're telling me you can create a whole generation of people where they don't have a certain trait, but that means any trait you can take away. You can take away their height. You can make everyone short, but 2 people in the entire world, or you try to make everyone 5'5", or you make everyone 110 pounds.
Or get to it. Make everybody white.
Make everybody yellow,
Yellow,
red, orange.
red.
Maybe they want to create a new
Whatever
color.
superpower took a hold of this technology and used it for ill gains, yeah, the end result would be editing people out of history, editing whole groups of people out.
Saying, hey, this is a vaccination that you have to have or is required, and then you end up going real far. So now let's take it all the way down. Let's see how CRISPR works. We'll dumb it down as far as we can.
I mean, all of these videos, we could have sat here and tried to talk to you about this stuff, but we're presenting all the evidence here. There's more than just videos. You can go to Wikipedia, you can go to all these websites. You're going to see the same terms being thrown at you in different structures of the sentence. This is what we can find to explain what CRISPR is. It is dumbing it down, and we don't want to misinform or we don't want to lead you wrong. These are the best videos. They're objective. They're coming from both sides, the good, the bad, the ugly of CRISPR. It was important enough that we did all this research. We barely understand it. We would like to share it with you. That's what all this is about. Every tab up there is a different way to view this. It's the only way to view this subject because I'm sure most people don't know what CRISPR is.
Yeah, true.
We joke about it and all this other stuff, try to be funny, but in reality, it's serious. It has big implications, unseen. They could be happening now with obviously genetically modified food, or we're an organism ourselves, so genetically modifying us.
We start talking about designer
They're
babies.
eliminating stuff. They're trying to go in and eliminate stuff. Well, what if my skin color and my eye color is to be eliminated? That's
What if it's something that you got they want to get rid of so people can't reproduce in a certain way, or a
Right.
gene you have? They might say, oh, that gene's bad. In 10 years, that gene was gene we needed to save everyone, but we destroyed it. We can get, yes, we may take away a whole generation of disease, but we may also cause the master disease that can kill everything, the unfightable disease, the invincible disease, because it's like all these things are being edited, something adapts, just like with our medication. We take medicine. Medicine is almost like CRISPR. They make a medicine, they give it to a whole bunch of people, they see how it does. Oh, they note the adverse effects and reactions, and then they say, oh, here's version 2. You try this and see if it's doing any better to the same patient.
Yeah, I talked about before where some people's blood make them immune to certain stuff, and if we eliminate that group of people and then that disease comes around in mass,
right,
no one will have any natural immunity to it. The variations in races and DNA are important for all of human survival.
Yes.
That's why this is a very important subject.
So we're going to give it to you how CRISPR works in 2 minutes. This is the best we found that can explain it in layman's terms, because some of this technology, even they will say themselves, is untested. So they just got to human trials in certain parts of the world. It's not even worldwide known that there's human trials going on. So 2 minutes, we'll give it to you. Then we're going to break down the questions we've got, the things we're thinking about, the implications as far as women and their rights goes, men and their rights goes, your children goes. And listen, we're talking about in vitro fertilizations. What if you don't even need women to make babies anymore? You just need eggs. I want a baby now. All you need is my DNA and an egg, and then I can pick my partner's DNA. I can go to some girl, get her DNA, and then we can edit what they look at both our DNAs and say your child is going to be susceptible to cancer, spina bifida, this, that. I have them edit it out. We can put that baby in and have someone carry it in vitro. I have a baby designed by me tomorrow. But we'll get there. We'll start with the 2 minutes, guys.
This is DNA. It's the blueprint for making living things, from single-cell bacteria to plants to animals, including to humans. It influences almost every part of your physical being. The shape of your nose, the color of your eyes, sometimes your likelihood of getting certain diseases. But what if science could redraw your body's blueprint? What if it were possible to edit your DNA? Insert new sequences that change inherited traits, or remove genes that cause disease. Well, now it is more possible than ever with CRISPR. CRISPR is a combination of a scissor-like protein, such as one called Cas9, and a guide molecule. The molecule acts as a bloodhound, taking CRISPR on the hunt to specific sites within the genome. Once there, the protein cuts the cell's DNA on the spot. That completely disables the targeted gene. The cells then repair their DNA. They put in a new DNA sequence that the bloodhound also carried, or simply patch up the break caused by CRISPR's molecular scissors. Enter tiny cellular machines called ribosomes. They translate the edited blueprint, producing healthy proteins that the repaired genes code for. CRISPR's ability to repair may be most important at the beginning of life. Edit the genome of a very early embryo, one created by in vitro fertilization, and couples who carry disease-causing mutations may be able to have children who are spared from them. Once a child is born with a genetic disease, such as Huntington's or Tay-Sachs, it is more difficult to use CRISPR to treat it. It's also being used to research and test treatments for cancer, blindness, and liver disease, among others. But CRISPR isn't just for humans. Scientists are already editing mosquitoes' genomes, hoping to stop the transmission of deadly diseases like malaria. And the potential of engineering microbes may open the door to a new world of medical and technological advances. When you can edit DNA's blueprint, the possibilities are vast.
Wow.
Yeah, 2 minutes or less.
2 minutes or less. That's what we're talking about, people.
If you can't get it after that, I mean, we got someone who can really explain it in depth, but yeah, trying to simplify the subject is our best bet here.
It is.
You can make all the assumptions after watching all that we watched, but I'm sure the conclusion will be somewhere in the same ballpark. It has good, it has bad, and it seems like because we have it now and we've already taken advantage of the good, it's pretty much just some bad stuff left really to happen. So I don't want to be that guy, that person, but you know.
So we're gonna break it down, and we're going even simple.
Yeah.
They've been buzzing about CRISPR, is what they're saying. Like they say, from reviving the woolly mammoths, a promise of distant cures for cancer, to the unproven potential for genome editing, stretched far and wide. I mean, like they said, you can have a baby free of disease that's designed in the womb, basically. You know what I mean? Or in vitro fertilization, edit genetic code, and they literally implant it. So, you know what I mean? So it's piqued the interest of many. and we're going to take a closer look. So it says, what is CRISPR? CRISPR, or more precisely, CRISPR-Cas9, which is the CRISPR-assisted, wait. Yeah, CRISPR, I forgot what, see, that's the words, man.
I can take it. It's a molecular toolkit that scientists have developed to make precise edits to the DNA, or our code of life. Actually, it was borrowed from bacteria, where in its original form it was used to protect microbes from attack by viruses. You can consider yourself, I guess, a bacteria, and
I'm a bacteria.
The undesired traits or whatever inside of us is what it's trying to protect us from. I guess if that makes sense.
It's like, how does it work? It says the system is made up of two parts. The first strand of RNA, DNA's chemical cousin, which matches up with the region of DNA inside a cell that a scientist may want to target. So we're talking about scientists targeting specific things.
Yeah.
It's not just, oh, we get a CRISPR. It's not just like like a CRISPR is not like a CRISPR. It's
like a natural happening.
Yeah, it's not a natural happening. CRISPR is a thing they're using. It's a tool. It's something to edit something else. That's what CRISPR itself is. And it says, then it acts like a shepherd to guide to the second component, a pair of molecular scissors called Cas9, to the site of the action where it makes a snip across the DNA. And what they mean by snip, basically, it gets rid of the bad part. The scissors are the analogy of basically telling you how it works. It's a lot probably more technical than that. How you probably snip DNA and repair it. I'm not gonna lie, way beyond my understanding. But they're trying to equate it to like that's what it does. It removes it, gets rid of it, snips it, cuts it, destroys it. And we'll get into the destroyed part too, because we're thinking these things. It's cutting this DNA out and it's repairing. But what's happening to things that are destroyed? What's happening to if maybe it destroys the wrong thing? It says, what can it do? Our scientists have chopped up their target region of DNA. A number of possibilities are opened up. It's like they could disrupt the function of a particular gene, like I was saying, cut it out. They can make precise spelling changes to the sequence, the DNA sequence, or slip an entirely new one, like they're saying. So they could say that's where we're talking about. There's 3 DNAs from 3 people. So they took the mother's DNA, the father's DNA, and there's somebody who had DNA that could fix whatever the thing was. Right. And that's why they put the 3 DNAs together, because the 3 actually made one perfect one, or one that they deemed perfect at the time.
Well, allow them to give birth to that child without, with a defect or a disability or something.
And they say they say it's a precise method of modification. I mean, you got to be precise. We're talking about human genetics here. You know what I mean? One wrong thing creates an extra arm or an extra leg, or
if someone's born with cleft palate, or their heart outside their body, or their nose is gone. They went to change the color of the eye, and what they actually did was clip away their nose and their ears.
So they're saying, is it worth the hype? And it says this CRISPR allows scientists to edit DNA in a way that's quicker, cheaper. Cheaper's not always better. I'm going to put that one out there. And more accurate than ever before. So it's an exciting development, opening up new possibilities for scientists across the globe, working in a number of different fields. And we see the good. We see how it can help people. But there's always a downside. But it says, but looking beyond the lab, it's still in its very early days. Even now, it's farther beyond this article, but it's still early for this type of technology. We just discovered DNA 70 years ago. Now we're playing with how we make people.
Yeah.
So it's like, it ends up being one of those things where take this with a grain of salt. And this technology may not be, even though it's cheaper, may not be accessible to the average public anytime soon. You know what I'm saying? So then we start looking at, well, it's great, but it's like, there's concerns. You know what I mean? There's concerns about what happens if it misses a target, which it can. That's what it says right here. It says DNA is complex and many genes are intricately linked. So cutting out this, like I said, may cause something else. So let's take a look at a video, somebody's description of what could happen when CRISPR doesn't go according to plan, because we've told you what it can do. We told you it can save lives. We told you get rid of diseases, cure the world. But we have yet to speak on what happens if it's bad. You know what I'm saying? So we'll go here and we'll take a look at this.
With all the hubbub about CRISPR in the news, it may feel like using it to pick your kid's eye color is just around the corner. But this seemingly miraculous gene editing technology may not actually be as simple or as safe as we thought. Just so we're all on the same page, CRISPR, or more accurately CRISPR-Cas9, works by cutting the DNA of a cell at a designated spot, say a section of DNA that represents a certain gene. then, when the cell tries to repair the damaged DNA, it often ends up just disabling the gene altogether. This has all kinds of uses. Maybe we want to turn off a gene that produces too much of a certain protein. CRISPR can do that. Say we want to see what a certain gene even does. CRISPR can help us do that too. Ever since it was first used to edit living cells in 2013, CRISPR has been touted as a promising technology for treating inherited disorders, cancer, and other diseases with no current treatment options. Teams of researchers around the globe had even hoped to begin human trials in 2018. But none of those trials have taken place yet, with the exception of China, where researchers have been working with CRISPR in humans since 2016. But no results from these trials have been published so far.
Chinese.
And in the US, the FDA just placed a clinical hold on the first proposed gene editing human trial. But why? Several new studies released in the last few months suggest that we need to be more cautious when editing the human genome. Two of these studies found that when CRISPR performs its hallmark trick and cuts DNA, that damage can kill the cell or make it stop growing. CRISPR modifications are also less likely to kill cells that have a defective version of a gene called p53. p53 plays a role in preventing the onset of cancer by regulating a cell's life cycle. So by leaving more of the defective cells alive than healthy ones, CRISPR may be inadvertently raising the risk of cancer in that patient, which is like the opposite of the goal. And we haven't even gotten to the most recent study that raises concerns. Up until now, CRISPR-Cas9's cutting function has been accurate in the specific area of interest, the spot in the DNA that's supposed to be cut. But that's because researchers were only looking for mutations caused by CRISPR in the immediate vicinity of the cut. New research reveals that in about 20% of cells, CRISPR results in much larger deletions than we thought, up to more than 100 base pairs. Researchers didn't notice this before because they were looking for harmful mutations and didn't see any, but that's because the entire region was gone. In CRISPR treatments that would target billions of cells inside the human body, this could lead to, again, a risk of cancer, putting us right back at square one. So the human trials have been delayed. Where do we go from here? CRISPR permanently alters your genome, so we want to make sure we get it right before we make moves in real human bodies. Well, science marches on. New and improved versions of this kind of technology are already racing forward, like a genetic editing tool called Repair, which uses a different cutting enzyme, Cas13. Repair works with RNA instead of DNA to make temporary or reversible changes. There's also a newly engineered protein that can alter individual base pairs rather than a whole chunk of DNA, acting more like surgical forceps than a pair of pigeon scissors. So continued progress and exciting breakthroughs in this field are still happening. We're not at the end of our CRISPR rope yet. It's just that we need to take a step back from the hype and carefully analyze what's really going to be safe as we move forward. To stay current with all the CRISPR news that's sure to be plentiful in the coming years, subscribe to Seeker. And for more on how your body, specifically your immune system, interacts with the technology, check out this video from Julian. And fun fact, CRISPR doesn't just present possibilities in human biology. It could be used to genetically engineer crops, too. I'm Marin. Thanks for watching Seeker.
Yeah, Marin.
Marin. You said her name's Marin.
Marin, yeah.
So the hype, you know what I'm saying? They're saying like she said, is it worth the hype? That's what we're just looking at.
There's risk.
There's risk, and then we're looking. She mentioned something interesting, cancer. I know something that a lot of America and people all over the world dealing with. And it says cancer is caused by faulty genes. So recreating these in the lab with CRISPR allows researchers to explore the underlying biology of disease and understand more about how it develops. Now, that is one good thing about CRISPR as well. And it says that's what their scientists are doing for a type of brain tumor called, glioblastoma? Something like that. Glioblastoma or glioblastoma.
You guys see it.
Yeah, I can't.
Type it into your search engine. Do your research.
Right. Do your research. It says tweaking genes in cancer cells could also help identify those that are essential for the cell's survival and therefore could be targeted with new treatments. On top of that, scientists could use the technique to explore ways that cancer cells become resistant to drugs, potentially opening up new ways to stop this from happening. Now, obviously, can it cure cancer? They're saying cancer isn't a single disease. It's more than a group of 200 unique diseases. So there's no one size fits all. They say it's possible.
Possibility in the realm of possibility.
Promising, possibility. These are the words the medical field they say to make you feel good when they've really got nowhere.
Well, they want more money to fund this type of research.
This is promising. They said the most promising idea so far is to use it in cell therapy for cancer patients, where the patient's own immune cells will be taken out of their body and tweaked, and then giving them a power up so they can better attack the cancer when it was given back to the patient. But that idea still needs testing in clinical trials. They're saying, what can't it do? It says, with the advent of gene editing came the idea this could be potentially used to correct faulty, disease-causing genes in people, therefore curing their illness. This remains a long way off and would be incredibly complex area to study, but it's not impossible, which I believe, because we've got this far,
Right.
which means we can definitely get farther.
Less than 100 years we're here. So.
Right. It says much more research is needed first, and where the desired edits might involve correcting inherited faulty genes, there are huge ethical questions to address, particularly around editing human embryos. These kinds of public debates, discussions, or expert recommendations are already underway. And then it says, here's number 9. So where are we now? In the context of cancer, CRISPR is the beginning to move from lab bench to bedside. Last year, scientists in China began trialing CRISPR-edited immune cells in lung cancer patients, which you saw her say in the video. But as of we know, there's no results have been published from China regarding that study. I find that very interesting as well. China does like to boast about their smarts a lot.
Well, why would they want to keep this?
And why would they want to keep this a secret? So you never know. You never know. And then, brings us over to genome editing. And that's where we're at. Genome editing and immunotherapy. And it goes more than just genome editing and immunotherapy. It goes to food. Like she says, it goes to animals. What about when they're using CRISPR? You never know. 8 months from now, they're using CRISPR in animals. And then all the cows die. Yeah. And nobody knows why.
Tinkering too much.
Or we just happen to notice that there's like a whole bunch of the same animal dying across America, and it's because they've been feeding it something, or they went and, this group of chickens came from a genetically modified CRISPR lab.
Let's say that they cycle through livestock that breed together, and they eliminated this one livestock group A. We've eliminated some things in them so that they live longer, they have better immunity. But when we're breeding them with livestock from lot C or D or E, their genetic offspring will have a lessened immunity, and they won't be able to fight a certain disease, and then they become riddled with it, and then we start consuming that livestock, and it starts altering us in negative ways. There's like so many implications when we start talking about using this stuff to alter DNA.
I mean, we saw what hormones have done just in food to the children over the last 20 years.
And that's within this exact realm. Hormones are just a way to manipulate aspects of the body and create a different outcome. And that's what they're doing. Now they're altering on a molecular level, a much smaller level, your DNA. Instead of regulating or irregulating a hormone in your body and a gland, now they're doing it to your DNA. This is
the future.
Yeah, this is our future. We're constantly trying to improve ourselves, work on ourselves, and hopefully we don't kill ourselves in the process.
One thing about CRISPR, though, and as we come to this, the cancer research from the UK.org, news.cancerresearch, it went out of the lab and into a patient. Genome editing techniques have been used in people to treat cancer and other diseases before, and there was lots of excitement when news broke in 2015 of a 1-year-old girl with acute lymphoblastic leukemia, ALL, like ALS, ALL. We have the ALS challenge and all that here in America. Being treated with a similar editing technique known as TALENTS after all the other treatments had failed for her leukemia. She received a transplant of cancer-fighting immune T cells from a donor, which had been tweaked in the lab to give them 2 new characteristics. Normally, the donated cells would see their new environment as foreign and attack the patient's healthy cells, but genes that control this process were turned off with, obviously, the genetic editing. It says the T cells will also be susceptible to attack from anti-cancer drugs that the baby was receiving, so modifications were made to protect them. They said she responded well to the treatment, and another infant received similar therapy. So it's like that's a prime example where they have used it already. And then it's like they're talking about late last year, a Chinese group became, and this would have been 2014, the first to use CRISPR-edited cells in humans. And then they said the team took immune cells from a patient with aggressive lung cancer and edited them in the lab. This editing deactivates a gene that allows tumors to put the brakes on these immune cells, preventing them from attacking cancer cells. by switching off the gene, which produces a molecule on the surface called PD-1. Is that like P53 or whatever? It says the force of the body's immune system is released, helping it clear the tumor. The drugs that targeted PD-1 are among the much lauded immunotherapy treatments already showing promise in advanced melanoma and lung cancers. So with all these big words, they're saying that the gene editing therapy is working. And then it says by switching off the gene, by switching something on or off in your body sometimes creates a reaction or an adverse reaction. And that's what they're talking about. Sometimes you have to deal with down the line, like your body may start to create a protein that it didn't before because you turned off something. And now that's the other things you have to look at as far as the sides go. It's like you might be able to cure this, but in 10 years, is this person going to have tumors because...
Or anything. Osteoporosis could be the result of doing stuff like this. Targeting something in a woman's body, something in anybody's body. And every disease really could be the implication of some experiment before that the public knows nothing about. So try to keep that in mind. It's been 100 years. I could think of a bunch of stuff that just popped up. I don't know how long we've been using the CRISPR technology, but...
Since probably 2012.
2016, he said?
I think they were playing with it in 2013. Remember this when they were in the lab? They said in 2013 CRISPR. So I'm saying 2012, 2013. They were recognized by the Nobel Prize in Chemistry in 2020, which was just 2 years ago. You know what I mean? And now when we go back to looking at CRISPR, it begins to make a little bit more sense when we go back to where we started at the beginning about what's CRISPR and what's Cas9. Now when we say it's a family of DNA sequences found in the genomes, now we know. You know, we know this, like, it's found in our bodies. We know that our cells produce this, so we know our cells use this to fight things. It's like, now it makes more sense. The big words come down to be a little bit more. We know that CRISPR is the tool that is used to fight these things.
Scientists have figured out a way to hijack your body to fight other aspects of your body, the aspects we don't desire.
The aspects that we do not desire.
Yeah, that's it's scary phrasing it like that because there are, once again, there's parts of people that others don't desire to see, and you could just edit whole groups of people out of existence, like a painting.
Yes, for sure.
Yeah, it's just like it's that easy. This technology could take off. People could say it's an inherent right, and they can use it to do whatever they want with it because it's their bodies. It comes from them. They're going to use it in themselves. Man, like 23andMe, people will have access to this technology in their home one day.
Time to do.
Okay, and then we got where we were breaking that down to and where we're coming to. Now we got that.
This has now changed with a revolutionary new technology now entering the stage, CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work?
Eternal youth? Nah, we didn't even talk about eternal youth.
Bacteria and viruses have been fighting since the dawn of life.
Okay.
So-called bacteriophages, or phages, hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell, microorganisms, plants, animals or humans. But despite the revolution CRISPR is for science, it's still just a first-generation tool. More precise tools are already being created and used as we speak. In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients in the lab, proving that it was possible. Only about a year later, they carried out a larger-scale project with rats that had the HIV virus in basically all of their body cells. By simply injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from cells all over the body. In a few decades, a CRISPR therapy might cure HIV. And other retroviruses, viruses that hide inside human DNA like herpes, could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever. But all of these medical applications have one thing in common. They are limited to the individual and die with them, except if you use them on reproductive cells or very early embryos. But CRISPR can and probably will be used for much more. The creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene pool.
Yeah, that's what I would be worried about. Now here we are, designer babies.
The means to edit the genome of a human embryo already exist, though the technology is still in its early stages. But it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore. Early on, vanity traits will mostly be left alone. But as genetic modification becomes more accepted and our knowledge of our genetic code enhances, the temptation will grow. If you make your offspring immune to Alzheimer, why not also give them an enhanced metabolism? Why not throw in perfect eyesight? How about height or muscular structure? Full hair? How about giving your child the gift of extraordinary intelligence? Huge changes are made as a result of the personal decisions of millions of individuals that accumulate. This is a slippery slope. Modified humans could become the new standard. But as engineering becomes more normal and our knowledge improves, we could solve the single biggest mortality risk factor, aging. 2/3 of the 150,000 people who die today will die of age-related causes. Currently, we think aging is caused by the accumulation of damage to our cells, like DNA breaks and the systems responsible for fixing those wearing off over time. But there are also genes that directly affect aging. A combination of genetic engineering and other therapy could stop or slow down aging, maybe even reverse it. We know from nature that there are animals immune to aging. Maybe we could even borrow a few genes for ourselves. Some scientists even think biological aging could be something that eventually just stops being a thing. We would still die at some point, but instead of doing so in hospitals at age 90, we might be able to spend a few thousand years with our loved ones. Research into this is in its infancy. And many scientists are rightly skeptical about the end of aging. The challenges are enormous, and maybe it is unachievable. But it is conceivable that people alive today might be the first to profit from effective anti-aging therapy. All we might need is for someone to convince a smart billionaire to make it their next problem to solve. On a bigger scale, we certainly could solve many problems by having a modified population. Engineered humans might be better equipped to cope with high-energy food, eliminating many diseases of civilization like obesity. In possession of a modified immune system, with a library of potential threats, we might become immune to most diseases that haunt us today. Even further into the future, we could engineer humans to be equipped for extended space travel and to cope with different conditions on other planets, which would be extremely helpful in keeping us alive in our hostile universe.
She's got web feet. A few grains of salt. Here we go. Nothing wrong.
Still,
with
a
progress.
few major challenges await us, some technological, some ethical. Many of you watching will feel uncomfortable and fear that we will create a world in which we will reject non-perfect humans and pre-select features and qualities based on our idea of what's healthy. The thing is, we are already living in this world. Tests for dozens of genetic diseases or complications have become standard for pregnant women in much of the world. Often, the mere suspicion of a genetic defect can lead to the end of a pregnancy.
I thought I said barbecue syndrome with the thing.
Take Down syndrome, for example, one of the most common genetic defects. In Europe, about 92% of all pregnancies where it's detected are terminated. The decision to terminate a pregnancy is incredibly personal, but it's important to acknowledge the reality that we are pre-selecting humans based on medical conditions. There is also no use in pretending this will change, so we have to act carefully and respectfully as we advance the technology and can make more and more selections. But none of this will happen soon. As powerful as CRISPR is, and it is, it's not infallible yet. Wrong edits still happen, as well as unknown errors that can occur anywhere in the DNA and might go unnoticed. The gene edit might achieve the desired result, disabling a disease, but also might accidentally trigger unwanted changes. We just don't know enough yet about the complex interplay of our genes to avoid unpredictable consequences. Working on accuracy and monitoring methods is a major concern as the first human trials begin. And since we've discussed a possible positive future, there are darker visions too. Imagine what a state like North Korea could do if they embraced genetic engineering. Could a state cement its rule forever by forcing gene editing on their subjects? What would stop a totalitarian regime from engineering an army of modified super soldiers? It is doable in theory.
Trying to do that for all the
Scenarios like this
history.
one are far, far off into the future, if they ever become possible at all. But the basic proof of concept for genetic engineering like this already exists today. The technology really is that powerful. While this might be a tempting reason to ban genetic editing and related research, that would certainly be a mistake. Banning human genetic engineering would only lead to the science wandering off to a place with jurisdiction and rules that we're uncomfortable with. Only by participating can we make sure that further research is guided by caution, reason, oversight, and transparency.
Oversight. Who's the oversight committee? See, then that's where, like,
Do you
the
feel
government-
uncomfortable
the
now?
government?
Most of us have something wrong with them. In the future that lies ahead of us, would we have been allowed to exist? The technology is certainly a bit scary, but we have a lot to gain, and genetic engineering might just be a step in the natural evolution of intelligent species in the universe. We might end disease. We could extend our life expectancy by centuries and travel to the stars. There's no need to think small when it comes to this topic. Whatever your opinion on genetic engineering, the future is approaching no matter what. What has been insane science fiction is about to become our new reality, a reality full of opportunities and challenges. Videos like this would not be possible without viewer donations on patreon.com. If you want to support us explaining complicated stuff and maybe get your own bird in return, you can do so here. If you want to learn more about CRISPR, we put the sources and further reading in the description. More videos about the whole topic area will follow. If you want to be notified when it happens, you can follow us here.
See, the channels are here. I can support discussions in social media. The Curse Got Voices from Steve Taylor. Soundtracks by Epic Mountain. We're looking good here, people. You need to all of this information. Genome editing and all this stuff is what we were here to talk to you about today, people. Today. What we're talking about is CRISPR. That's what we're talking about. And we're coming to the end of our time here, people. We appreciate you stopping by for another episode of Reaction Time with Dave. Once again, I have with me Jack Phelps. That was us talking about CRISPR. What is it? Is it some chicken? Obviously, we found out it's not chicken. It's a gene editing tool in which you can use to edit DNA, not just in people, but in all organisms.
Right. Take out the undesirable, leave what is left to be desired, and no one really knows what the implications are. There's some good, there's some bad.
The future is to be seen. You know, research is always going forward. We just hope and we pray that things happen in the best way for us. You know, designer babies or something that can come out of this. Maybe we're looking past the small. We didn't even start with designer dogs. People are already breeding dogs with other dogs to make certain dogs. But what if we don't got to breed the perfect pit bull anymore? I can genetically engineer that perfect pit bull who's going to be 400 pounds, 5 foot tall. You know what I'm saying? Like when standing like straight up. I mean, that's possible. We were looking at babies, but dogs, they're messing with mice. What about dogs? You know what I'm saying? That's a possible thing.
They went straight to humans from mice, though.
I know, right? They didn't even say let's make special dogs. Let's make blue-haired dogs.
Doing dogs and doing food is very simple. Obviously, they could do things like this to some degree, and they call it intelligent design. You've seen some of the dogs; they're not that intelligent on the design. This is in our everyday life. And people are just overlooking what CRISPR is. CRISPR, specifically Cas9. It is something worth looking into. We could have did anything. We could have talked about anything. This came up. It was confusing but interesting enough to read because the implications in your everyday life are real.
Right.
They're real.
They are really,
the good,
really
the bad,
real.
and the ugly. We just presented all of that.
We try to present it all to you. We try to let you guys get your own opinion, hear a little bit of ours, and also see facts. So we're coming to the end of our time. We would like to say thank you to all of you for stopping by and enjoy your day. Check out those credits. Those credits. Have a good day, everybody.